Some 30 years ago, when I was a young research student in Cambridge, I had the great good fortune to have as my tutor and as my advisor Dr. F.H.A. Marshall, who was then Dean of Christ's College in Cambridge. Marshall had then, for many years, been studying the effect of environmental stimuli, the amount of food in the diet, the effect of daylight, length of daylight, the effect of temperature, effect of presence of mates, on the sexual periodicity and sex cycles. And Marshall suggested to myself that as my first research project, I might attempt to stimulate the brain in rabbits to see if in this way any evidence could be obtained that brain stimulation affected the state of the ovary. Now, if I might turn to this model, this depicts the base of the brain, the hypothalamus, connected to the pituitary gland by means of this stalk. Here is the pituitary gland, and the anterior part of this gland secreting the two hormones, follicle-stimulating hormone and luteinizing hormone. One of the first problems, of course, was how to stimulate this part of the brain, which is at the base, lying on the base of the skull. And for this purpose, we devised this stereotaxic machine. This is part of an old microscope that we found in the basement of the department. This we constructed. It consists of little clamps for orientating, fixing, and orientating the head of the fully anesthetized rabbit. And then by means of this rack and pinion, it was possible to lower electrodes into the hypothalamus, to apply electrical stimulation to this part of the brain, and then some 10 to 20 hours later to see whether the animal had ovulated. And the quite clear-cut upshot of this experiment was that electrical stimulation applied to the brain would cause release of luteinizing hormone from the anterior part of the pituitary gland, and this would in turn result in ovulation some 10, 15 hours later. The next immediate question, if we turn this model round, we can see what the view is on a section, a midline section through the hypothalamus here, through the pituitary stalk, and the anterior lobe of the pituitary gland, which secretes the luteinizing hormone. And here on this model, one can see depicted a great wealth of nerve fibers that run from the brain down into the gland, but they all end in this posterior part, with which we are not concerned, and very few, if any at all, come through into this anterior part. So the problem then was simply how, through what mechanism does the brain influence this gland? It clearly does influence it, but what is the connecting structure through which the influence is mediated? In 1930, 1933, two papers were published by Professor Popar, Romanian professor of anatomy, working with Dr. Una Fielding in University College London, and they described for the first time a little system of blood vessels starting here in the hypothalamic area, passing down the pituitary stalk, and breaking up again here in the anterior lobe of the gland. They describe this working on the human, and in this photograph over here, we have a view such as they must have seen in their early studies. This is a preparation published by Professor Daniel and his co-workers, showing the hypothalamic region in the human up above. This is the pituitary stalk of the human with the pituitary gland below here. And it's quite clear to see that there is a profuse system of vessels running down the pituitary stalk, breaking up into small vessels up in the hypothalamic region, and breaking up into small vessels down below in the pituitary gland. So here, anatomically, was a system, a little connecting link, that could possibly be responsible for mediating the influence that the brain exerts over the gland. Now, clearly, this would be a very basic function in reproduction and the preservation of the species, and in that case, one would expect these vessels to be present in a whole variety of vertebrates, mammals, birds, and the lower vertebrates. So this was investigated, and here, for example, is a section through the same structures in a bird. Here is the hypothalamic area of the brain. Here is the pituitary gland below, and in this case, the blood vessels have been injected with Indian ink so that they show up as the black structures. And one can see rather beautifully here, almost as it were a collecting cup of vessels, and then a redistribution of these structures in the gland. And further investigations showed, in fact, that these structures were present in all forms from fishes up to the human. Now, in this photograph, we are looking at the brain upside down. Here is the front end, two large nerve trunks passing forwards, and here, right under the middle of the brain, is the pituitary gland with the little stalk passing forwards and upwards to join the hypothalamus. And the technique that was worked out to cut the pituitary stalk was to approach it from the side, to lift up the lateral part of the brain with an elevator here, approach the midline structures. Here is the carotid artery, here the hypothalamus, and here, running from the hypothalamus down to the pituitary gland, is this little, in this diagram, black pituitary stalk. And having exposed the region so, one could then cut the stalk and ascertain what happened to the activity of the gland when the animal recovered. I think this shows up rather more clearly in this diagram. Here we have the hypothalamic region. Here is the pituitary stalk with the portal vessels running from the hypothalamus down to the pituitary gland. And here this arrow depicts the secretion of the follicle-stimulating hormone and the luteinizing hormone from this anterior lobe. Now, cutting the pituitary stalk, so sectioning it, cutting it across here, resulted in only slight delays in the sex cycle, and on investigating this area under the microscope, we found that in many cases these vessels had regenerated and regrown across the site of the cut. So a second type of experiment was devised in which after cutting the stalk, a little plate of wax paper was inserted between the cut ends to prevent any regeneration. And these animals behave quite differently. If such regeneration was prevented by this plate, then the activity of this gland was largely lost. The secretion of follicle-stimulating hormone and luteinizing hormone no longer occurred. The reproductive processes ceased, and on examining the reproductive organs, they were found to be atrophic or wasted and degenerate. So this fitted in with the general idea: if the gland receives this particular blood supply, it becomes active. If this particular blood supply is permanently interrupted, then the gland loses its control of the reproductive organs. A second type of experiment shown in this diagram. Here again we have the hypothalamic area of the pituitary stalk, here the anterior lobe of the pituitary gland. Now, in this experiment, in which the pituitary gland was transplanted, first of all the whole gland was removed, and then a fragment of the removed gland was taken and put not back into its original capsule, but placed, for example, here. This was an experiment in collaboration with Dr. Jacobson of Sweden, and we found that if the transplant was made just in this region, that these little vessels would grow from, in this rather unusual course, to the transplanted gland, rather than regenerating back down to the site of the original gland. And in these animals, reproductive cycles recur. They would mate with male animals, they would become pregnant, they would deliver a litter, and they would raise their litter. On the other hand, though, if the gland was placed up here, so that these little vessels couldn't reach this tissue would receive a blood supply, but from other vessels, but would then remain without very much sign of function. It would remain anatomically present, but would not be active, and the reproductive organs would again waste and atrophy. These experiments then showed quite clearly that this particular set of vessels was of fundamental importance for pituitary gland function, but of course it didn't show in what way they acted. Now, at this stage, several theories were put forward. One, if we take this diagram, this again is supposed to represent the hypothalamus, the anterior pituitary gland, hormones being secreted here. One view concerning how these vessels acted was that they just regulated the total amount of blood being received by the anterior pituitary. If they carried more blood to the gland, it became more active and secreted more hormones to act upon the ovary. If the vessels cut down the total blood supply, the gland secreted less hormone and the ovaries became inactive. This was a view not held by many people, since the pituitary gland here, this anterior lobe, secretes something like 6 hormones, and this would be a rather gross way of regulating the very delicate mechanisms concerned in glandular function. A second theory that I think most workers felt more likely to be correct is shown in this diagram. These black lines are supposed to depict nerve tracts coming from the hypothalamus, with the nerve terminals ending at the upper end of these vessels. The vessels then carrying blood down, from which the nerve terminals have liberated various chemical substances. These chemical substances are carried down to affect the activity of the gland cells. In other words, a sort of neurosecretion, a secretion from the nerve tissue being transmitted into the portal vessels, and these substances acting on the gland. The neurosecretory material is nowadays referred to in terms of releasing factors, that the nerve fibers are thought to release these various factors that act upon the gland. In order to substantiate or disprove such a view, it became important to know the detailed connections here between the nerve terminals and the upper end of these vessels. And here we see an electron microscope photograph of just this region. This, on the right upper corner, is the wall of a blood vessel. This is the inside of the blood vessel. Here are nerve terminals. This whole oval structure here is the end of a nerve fiber, the end of another nerve fiber here, one over there. And one can see that in these nerve endings, there are these little black masses that are suggestive of neurosecretory material. That is, that this nerve terminal might liberate the contents of these little vesicles, which might then be transported across into the blood vessel, which would carry the releasing factor down into the gland. Now here, for myself, began the most exciting part of the work, an attempt to extract from brain tissue these chemical substances and then apply them to the pituitary gland in some way or another to see if these more purified chemical fractions could alter the function of the gland. And I think it's true to say that the evidence quite strongly points to there being 6 different types of releasing factors, each one concerned with the control of the secretion of one of the 6 anterior pituitary hormones. And at this point, a great deal depends upon the skill of the chemists in purifying these extracts and finally establishing their structure.